Now, moving towards the topics which we are going to discuss in this lecture, so they include the raw materials for the fermentation process, as a result of which we get the ethanol as a final product. Now, the conditions necessary for the fermentation process to carry on so that we can get the ethanol as a final product. Various methods for ethanol production. The general sketch of bioethanol production. Now moving towards the crucial steps in the biosynthesis of ethanol. So first is the addition of diammonium sulfate to the broth. So that is a very important step in the production of ethanol. Lowering pH. So normally the ethanol is produced at a low pH. So if in the fermentation tank the pH is increased, then the percent yield of ethanol will also be affected. Addition of fermenter in the form of yeast for the fermentation to take place. So yeast also has a very important role, and when the yeast is added as a starter culture in the inoculum in the form of a fermenter, so then the process of fermentation can initiate. Fractional distillation, so these are the steps which are taking place at the end of the run when you are harvesting the product, and fractional distillation is very important for getting the pure form of the ethanol. And the conversion of commercial alcohol to absolute alcohol. So as a result of the process of fermentation, the end product alcohol which you are getting in the form of ethanol is not very pure. So then there are certain steps which need to be taken to convert it into absolute alcohol, which is 99% pure. Now moving towards the introduction. So fermentation is a biochemical process that occurs in the presence of enzymes which are being secreted by microorganisms such as yeast or bacteria, and as a result of fermentation, carbon dioxide, ethanol, and energy are produced. Now, the alcoholic fermentation or biosynthesis of alcohols by fermentation is basically an anaerobic process, mostly carried out in the presence of yeast. So you can see that yeast has a very important role in the biosynthesis of ethanol by the process of fermentation. Now the alcohol which is obtained by the process of fermentation is 12% and not more than 15%, because beyond this the enzymes they become inactive. So when the enzymes are inactive, then the conversion of the ethanol cannot take place, and as a result no more ethanol can be produced. Now this technique is exclusively adapted to prepare ethanol on commercial scale by developing countries to combat energy crisis. So in order to use the ethanol for different types of uh, energy as a source of energy, so in developing countries These fermentation methods are used for the production of ethanol. Now, the raw materials and the fermenters which are used for the production of ethanol in the process of fermentation, and the types of fermenters which are employed, they include certain yeast and bacteria for the production of alcohol by the process of fermentation, and among yeasts, the Saccharomyces cerevisiae, and among bacteria, Zymomonas mobilis are used for the production of ethanol. Now, the raw materials, they are generally serving as a substrate for the alcoholic fermentation. So, the production of alcohol or ethanol by the process of fermentation is also referred as alcoholic fermentation, which also means the production of alcohol by the process of fermentation. Now, the sugary materials, they include molasses, whey, glucose, which are the source of energy and nutrients for the bacteria to grow and reproduce. Starchy materials are also used as raw materials. They may include potato and wheat, and cellulosic material like wood and bagasse, they are also generally used in the fermentation technique for the production of ethanol. Now, the conditions which are necessary for the fermentation process to go in a smooth manner for the production of ethanol, so they include the size of the fermenter tanks. So the size of the fermentation tanks is around 0.125 million gallons. So like around 0.1 million gallons of the culture can be accommodated into the tanks. So you can observe that how much big the size of the gallons is. The ideal pH is around 4 to 4.5. So that is very important, that to maintain the pH within the fermentation tank around 4 to 4.5, and the optimum temperature is in the range of 21 to 27 centigrade. So if the temperature is below or above this range, then the effect will be on the yield of the ethanol. The proper aeration is also required so that the oxygen can reach to the cells and so they can grow and produce. Absence of any preservative or contaminant. So the contaminant can be in the form of another bacteria or phage, which can kill the bacteria which are producing the ethanol, and as a result the whole process can become faulty. Now the preservatives, so they should not be added because these preservatives are normally chemicals and they can damage the metabolism of the bacterial cells. And the proper dilution is also very important during the process of fermentation. If you are using a fed batch method, so the dilution should be carried out by taking out the amount of the culture which has developed in the same manner, in the same amount of quantity as the amount of nutrients are being added to the fermentation tank. Now move towards the methods for ethanol production. So the first one is the wet milling process. The second one is dry milling or malting process by which the ethanol is being produced. Third one is dilute acid hydrolysis process. The fourth one is enzymatic fermentation. So all these processes are types of fermentation, but they differ either in using the batch mode or the continuous mode or the fed batch mode. So that is why they have different types of yields as a result of the final product. Now the enzymatic fermentation process is prevailed in the biochemical industries, and it is also preferred. Now the general sketch of the process is shown in this figure, in which you can see that first of all you have corn, which is subjected to the grinding step, then to the cooking step, and then to the liquefied state. Now when the corn has reached the liquefied stage, at that stage the enzymes and yeasts, they are added to the liquefied state of corn. Now the next step is to saccharify. So saccharification is to convert into a simple soluble fermentable sugar by hydrolyzing a sugar derivative or complex carbohydrate. Now after saccharification, the process of fermentation is started, and as a result of the complete run of the fermentation process, the primary products are obtained, which are carbon dioxide, and after fermentation by the refinery step, which is distillation, we get the ethanol as the final product. Now after distillation, the remaining material is subjected to centrifugation, and as a result of centrifugation you get two types of layers from the centrifugation. One are the distiller's grains, which are the lower layer, and the other are the distiller's solubles, which is the upper layer. Now the centrifugation step is followed by evaporate. So during the centrifugation step, the lower layer is being taken out and the upper layer is being subjected to evaporate, and as a result you get the distillers solubles. Now both of them are mixed together, and then they are used for different purposes. So this is the general sketch that how the ethanol production is taking place along with the step which is the saccharification of the liquefied corn, as well as the fermentation step and the refining steps, which include the distillation, after which you can get the ethanol. Now moving towards the pre-treatment of the raw materials. So it means that if you are going to use certain raw materials, do you need to treat them with certain chemicals or wash them or add certain other nutrients to them? So the sugary raw materials, they require no pretreatment, but the starch and cellulosic material, they do require the pretreatment. And as we discussed in the previous slide, that this includes the grinding, cooking for 2 to 3 hours, and then liquefying, and then ultimately the saccharification of the raw materials. Now the saccharification, which I explained in the previous slide, can be carried out in an acidic or enzymatic environment to release the monosaccharides, which are then the fermentable sugars from which the ultimate product of ethanol can be generated. Now, moving towards the important steps which are carried out during the fermentation process. So, first one is the dilution. So, dilution, the most commonly used raw materials for fermentations are sugars, which are used within the range of 10 to 17%, and the molasses, which are used within the range of 50 to 55%. Now, when the molasses are being used for the fermentation process to initiate and prosper this, so it needs 70 to 89% dilution, so that to make the fermentable sugars in the 15 to 17% range concentration. So it means that when you are diluting the molasses, so automatically you are diluting the sugars and they are reaching to a percentage within the range of 10 to 17%. Now the starchy and cellulosic materials, they first need to be ground, like grinding, and then they are diluted 5 times. So ultimately you have a 18% starchy materials concentration within the fermentation tank. Now moving towards the nitrogenous contents. So in case of ethanol production, the addition of diammonium sulfate to the broth is carried out, and the nitrogenous matters in molasses acts as food for yeast. Now, if the nitrogenous content is lower in amount or is it poor, then the ammonium sulfate or ammonium phosphate is also added to the broth so that they can serve as a food for yeast. Another thing is the lowering of the pH. For maintaining pH in between 4 to 4.5, the dilute sulfuric acid is added to the broth, and this highly favors the growth of yeast. Now, the role of enzymes is very important in the process of ethanol production via fermentation. And the process of fermentation, it starts as soon as the fermenter in the form of yeast is added to the fermentation tank. Now, the mixture is maintained at 26 centigrade for 2 to 3 days, and the enzymes like invertase, they convert the starch molecules to glucose and fructose, which are the monosaccharides. Now you can see that the invertase has a very important role, that it is converting the uh, polysaccharides into monosaccharides of carbohydrate molecules, and subsequently then the zymase enzyme is acting on the fructose and glucose molecules and converting them to ethanol and carbon dioxide. So you can see that if the zymase and invertase, they are not present, so then it is not possible to get the target ethanol from the process of fermentation. Now the frothing happens due to the formation of carbon dioxide, and it is being removed by flotation. Now the distillation, it is a very important step in the refining of the alcohol, and as we discussed its importance in the sketch diagram for the process of fermentation in the previous slides. So the fractional distillation, the fermented liquid is known as wash, and this wash contains 12% ethyl alcohol or ethanol. Now this wash is sent to fractionating column, where the blow steam converts ethanol to the vapor form. And then the condenser, it condenses it as the boiling point of ethanol is less than water, so the ethanol is collected first as a fraction. Now, as a result of this distillation and condensation process, we get 95% alcohol, which is commercially known as the rectified spirit. Now, refinement is the final step in getting the absolute alcohol, and it gives the final touches, finishing touches to the process. So it is actually the commercial alcohol conversion to absolute alcohol. So the absolute alcohol is 99% pure, and it is obtained by the process of redistilling the 95% rectified spirit in the presence of lime and calcium oxide. So what are the functions of lime is that it absorbs moisture from the alcohol ethanol, so converting and taking out the water molecules or moisture from the 95% alcohol and converting it into the 99% absolute alcohol. So these are the references, and for further reading and explanation you can consult these books.